This is Professor Will, CBMD, PhD. Today, Saturday, May 26, 2018. It is 2012 Eastern Standard Time. Biologist advocates ecological approach to improving human health. Date, May 25, 2018. Source, Oregon State University. Summary, chronic disease like cancer, autoimmune disorders, and obesity may ultimately vanish the efforts of medical intervention unless people change their diet, a biologist argues in a paper published this week. An important wider question is also raised by the study. Do mutations in titin predispose people to heart failure when exposed to other things that stress the heart, such as cancer drugs or certain viral infections? This is something we are actively seeking to address. More research is now needed to investigate how alcohol may affect people who carry the faulty titin gene, but do not have heart problems. Alcohol and the heart have a complicated relationship. While moderate levels may have benefits for heart health, too much can cause serious cardiac problems. This research suggests that in people with titin-related heart failure, alcohol may worsen the condition. Compared to DCM patients who didn't consume excess alcohol and whose condition wasn't caused by the faulty titin gene, excess alcohol was linked to a reduction in heart output by 30%. In the study, the team asked 716 patients with dilated cardiomyopathy how much alcohol they consumed. None of the patients consumed the high levels of alcohol needed to cause ACM. But the team found that in patients whose DCM was caused by the faulty titin gene, even moderately increased alcohol intake, defined as drinking above the weekly recommended limit of 14 units, affected the heart's pumping power. In the second part of the study, the researchers investigated whether alcohol may play a role in another type of heart failure called dilated cardiomyopathy, DCM. This condition causes the heart muscle to become stretched and thin. It has a number of causes, including viral infections and certain medications. The condition can also be genetic, and around 12% of cases of DCM are thought to be linked to faulty titin gene. He added that relatives of patients with ACM should receive assessment and heart scans, and in some cases have genetic tests to see if they unknowingly carry the faulty gene. Our research strongly suggests alcohol and genetics are interacting, and genetic predisposition to alcohol consumption can act together to lead to heart failure. At the moment, this condition is assumed to be simply due to too much alcohol, but this research suggests these patients should also be checked for a genetic cause. By asking about a family history and considering testing for a faulty titin gene as well as other genes linked to heart failure. These results suggest this condition is not simply the result of alcoholic poisoning, but arises from a generic predisposition and that other family members may be at risk too. The team found that the faulty titin gene may also play a role in the condition. In the study, 13.5% of patients were found to carry the mutation, much higher than the proportion of people who carry them in the general population. In the first part of the study, the team analyzed 141 patients with a type of heart failure called alcoholic cardiomyopathy, ACM. This condition is triggered by drinking more than 70 units a week, roughly 7 bottles of wine, for 5 years or more. In severe cases, the condition can be fatal, or leave patients requiring a heart transplant. Titin is crucial for maintaining the elasticity of the heart muscle, and faulty versions are linked to a type of heart failure called dilated cardiomyopathy. Now, new research suggests the faulty gene may interact with alcohol to accelerate heart failure in some patients with the gene, even if they only drink moderate amounts of alcohol. Good day, this is Professor Will, CBMD, PhD. Today is Saturday, May 26, 2018. It is 18:01 Eastern Standard Time. New link found between alcohol genes and heart failure. Date, May 25, 2018. Source, Imperial College of London. Summary, scientists have revealed a new link between alcohol, heart health, and our genes. Researchers investigated faulty versions of a gene called titin, which are carried by 1 in 100 people, or 600,000 people in the UK. For example, a person may feel ambivalent about initiating an intimate relationship because this challenges a more abstract goal of independence. The young adults in this study were aged 18 to 35 with an average age of 20. For example, a person's goal to spend more time with their family may conflict with their goal to get promoted at work. Ambivalence is thought to reflect a deeper motivational conflict of which the person is unaware. Attention to these deeper motivational conflicts may be an important step towards resolving them and relieving distress. However, when these goals generate conflict, they can contribute to psychological distress. Intergoal conflict occurs either because the objectives are incompatible or because pursuit of both goals draws upon a limited resource. such as time or money. People with poor mental health are more likely to report that their personal goals hinder one another. Such conflict between goals may be more manageable if it is conscious. However, ambivalence may indicate a clash between a goal and a higher order value that lies outside awareness. These were inter-goal conflict. When pursuing one goal makes it difficult to pursue another, and ambivalence, conflicting feelings about particular goals. The results showed that each of these forms of goal conflict was independently associated with anxious and depressive symptoms, but did not predict worsening of symptoms over the 1 month. This is Professor Will, CBMD, PhD. Today, Saturday, May 26, 2018. It is 17:56 Eastern Standard Time. Goal conflict linked to psychological distress. Date, May 25, 2018. University of Exeter. Summary of being torn apart, which personal goals to pursue is associated with symptoms of psychological distress, new research shows. Providing a link between empathy for pain and pro-social behavior at the neural level is crucial to understand our social human nature and target pharmacological approaches to treat pathologies in which these mechanisms dysfunction. For example, in a psychopath individual or in children with callous unemotional traits. An important social function for the tactile cortices. The results suggest that our tactile cortices, primarily evolved, perceive touch and pain on our body, have an important social function. They contribute to pro-social decision-making by helping to transform the sight of bodily harm into an accurate feeling for how much pain the victim experiences. This feeling is necessary to adapt our helping to the needs of others. Normally, participants gave more money when the victim experienced more pain. But when interfering with tactile activity, two related phenomena were observed. A, people became less able to perceive in how much pain the other person was, and B, they no longer adapted their donations as appropriately to the needs of the other. The researchers used electroencephalography, a method to record electrical activity of the brain, in human healthy participants. With this method, they found that the activity in tactile cortices increased when participants increased their donation. Later, they altered brain activity by using neuromodulation. To answer the question, researchers from the Social Brain Lab gave participants the opportunity to reduce the pain of a victim receiving a swat on her hand. They could reduce the pain by giving up money they could have otherwise taken home while their tactile brain activity was measured and altered. Helping other people in need is a foundation of our society. It is intuitive to believe that we help others because we empathically share their pain. Neuroscience shows that when we see somebody in pain, our brain activates tactile and emotional regions as if we ourselves were in pain. A study from Solijn, Netherlands Institute for Neuroscience, NA, investigated whether altering activity in these tactile brain regions while witnessing the pain of others would alter people's willingness to help. The results published on 08 May 2018 eLife are of great importance to understand our social human nature and to find treatments for pathologies like psychopath individuals. Good day, this is Professor Will, CBMD, PhD. Today, Saturday, May 26, 2018. It is 17:46 Eastern Standard Time. Which role does the brain play in pro-social behavior? Date, May 25, 2018. Source, Netherlands Institute for Neuroscience. Summary, the study suggests that our tactile cortices, primarily evolved to perceive touch and pain on our body, have an important social function. They contribute to pro-social decision-making by helping to transform the sight of bodily harm into an accurate feeling for how much pain the victim experiences. This feeling is necessary to adapt our helping to the needs of others. Another project has been able to show which proteins that interact with LRIG1 change its function. Of a total of around 20 proteins, it was shown that 4 proteins support LRIG1, but 8 proteins inhibit LRIG1. Previously, only one of these, about 20, was described. These are really exciting discoveries. You can actually talk about a breakthrough of significance for the field. However, the data are very preliminary and more future research projects are needed to elucidate the mechanisms. Scientists shown that the tumor inhibitory effect of the LRIG1 gene is partly related to the other proteins, so-called receptor tyrosine kinases, RTKs. These RTKs play a key role in the development of tumors, and they can contribute to several types of cancer if they are not inhibited. LRIG1 inhibits the migration of brain tumor cells partly through a specific RTK, MET, mesenchymal epithelial transition, the other studied gene. LRIG2 promotes the tumor by modulating downstream signaling to another RTK, PDGFR. FR. Scientists shown that the LRIG1 gene acts as a tumor suppressor. That is, the gene counteracts the formation of tumors in mice brains. On the other hand, the other studied gene, LRIG2, appears to promote malignant brain tumors. Scientists focus on the genes LRIG1 and LRIG2. They have their names of leucine-rich repeats and immunoglobulin-like domain proteins. The fact that these genes play a role in cancer is known since before, but their molecular mechanisms have so far been unclear. Several research groups around the world have for many years been trying to explain this mechanism. Understanding the importance of the genes to hold back malignant tumors can hopefully be of importance in the long term for cancer treatment. Good day, this is Professor Will, CBMD, PhD. Today is May 26, 2018 at 17:04 Eastern Standard Time. Genes of different importance for brain cancer. Date May 24, 2018. Summary. 2 genes play important roles for the formation of malignant cancer in the brain. One gene prevents the formation of the tumor while another gene promotes the tumor. We hope that these studies are done either in collaboration with our group or in the labs, and if the results are confirmed, that the pharmaceutical industry takes the lead in performing the trials to test the efficiency of the combined treatment in patients. From the point of view of applications, it is one of the most interesting findings of the study, as there are straightforward methods to measure chromosomal instability in tumors, but our findings have to be confirmed in a greater number of tumors from patients. Selected patients, the researchers also believe that they have found a possible explanation as to why 2 of the tumors analyzed did not respond to the treatment, allowing differentiation between patients who may benefit from those who will not. They observed that the effectiveness of the treatment was dependent on the degree of chromosomal instability of the tumor cells. The greater the instability, the more effective the treatment. Taxinus prevents cell division by damaging chromosomes and causing chromosomal instability. As suspected, given that p38 hinders this action, if we switch off the function of this protein in cells, they lose their protection and Taxinus can be more effective. The scientists used 9 tumors from patients which were grown in mice. In 7 of these tumors, including ER and triple negative, the p38 inhibitor strengthened, accelerated, or prolonged the anti-tumor effect of the Taxinus, blocking p38 to optimize chemotherapy. The researchers used p38 inhibitors to block the action of this protein in tumor cells. These inhibitors have already been used in clinical assays involving patients, but for other diseases. In this study, p38 inhibitors were combined with classical chemotherapeutical drugs called taxanes, such as paclitaxel and docetaxel. As expected, the cells treated with combined therapy showed greater accumulation of genetic instability, that is to say, they carried more DNA damage and a greater number of chromosome variations. Consequently, many of these cells died and the tumor mass shrank considerably. The experiments were performed in a mouse model characterized by development of triple-negative breast tumors. The kind of tumor accounts for 15% of breast cancer cases, and the only treatment to date consists of generic chemotherapy. In contrast, targeted treatments are available for the 2 other types. These discoveries could pave the way to improving treatment options for triple-negative breast tumors. Breast cancer is the most lethal tumor in women, causing 521,000 deaths worldwide each year. 6,213 of these in Spain. Sources: World Health Organization 2012 and SEOM 2017. Breast cancer can be classified into 3 types: estrogen receptor positive ER, ATR2 positive, or triple negative. The results demonstrate that the p38 alpha p38 from now on protein protects tumor cells from turning on DNA repair mechanism. In this regard, p38 safeguards tumor cells against the excessive accumulation of DNA errors or mutations. Tumor cells inherently tend to accumulate DNA damage, but in some this accumulation is greater, and we have observed that these cells are more dependent on p38 activity. Greater knowledge of the mechanisms that contribute to the survival of tumor cells is key to vanishing them. A study published today in the Journal of Cancer Cell Headed reveals a new protective mechanism for tumor cells in breast cancer and identifies novel therapeutical targets to treat this condition. The study has been funded by Competitive Advance and Proof of Concept grants from the European Research Council, ERC. Date: May 24, 2018. Source: Institute for Research in Biomedicine, IRB, Barcelona. Summary: p38 blockage has been shown to increase the death of tumor cells, thus causing tumors to shrink. The combination of p38 inhibitors with chemotherapeutical drugs Taxanes strengthens, accelerates, or prolongs the anti-tumor effect in patient-derived tumors grown in mice. Good day, this is Professor Will, CBMD, PhD. Today, Saturday, May 26, 2018. It is 16:49 Eastern Standard Time, tumor cells evade death through an extremist DNA repair. A study on breast cancer conducted at IRB Barcelona identifies the key role of P38 in safeguarding tumor cells against the excessive accumulation of DNA damage which would otherwise cause cell death. In the study, researchers used big data analysis to reconstruct the RNA thought to form global regulatory networks within breast tumor cells and their matched normal tissues. This approach identified the fact that 3 transposed UTRs are vital in regulating these global regulatory networks. Using this new information, they then disrupted these networks within breast cancer cells to test the effects on tumor growth. And researchers have historically thought that this was because 3 transposed UTR shortening induces the expression of proto-oncogenes, normal genes, when altered by mutation or expressed too high, become oncogenes that can transform a normal cell into a cancer cell. However, using a combination of computational approaches in cancer cell models, we found that 3 transposed UTR shortening in tumors actually causes tumor-suppressing genes to be turned off. Molecules throughout the body, the 3' untranslated region, or 3 transposed UTR, is a section of messenger RNA that can alter gene expression. It is known that shortening the RNA section promotes cancerous tumor growth. College of Medicine found that losing a section of messenger RNA that was previously thought to transform normal cells into cancerous ones actually acts by blocking a body's ability to suppress the formation of tumors. The finding could completely alter the way that medical science approaches the formation of tumors. Today's is Professor Will, CBMD, PhD. Today, Saturday, May 26, 2018. It is 16:22 Eastern Standard Time. Science News: new data changes the way scientists explain how cancer tumors develop. Date May 21st, 2018. University of Texas Medical Branch of Galveston. Summary: a collaborative research team has uncovered new information more accurately explains how cancerous tumors grow within the body. The study is currently available in Nature Genetics. These data highlight the relevance of combined proteomic genomic cellular screening to identify critical effectors of genes involved in the development of acute myeloid leukemia. Our results establish the novel role of SETD2 in the maintenance of genomic integrity during initiation and progression of MLL, rearranged AML, and contribute to further clarification of the molecular mechanisms driving MLL fusion-dependent leukemogenesis. Scientists carried the entire protein complexes around 7 different MLL fusion proteins in detail, which revealed 128 conserved interaction partners of MLL fusions, in further functional experiments that were carried out in collaboration with the IMP. Using genomic techniques including CRISPR-Cas9 genome editing, the researchers found that SETD2 loss caused induction of DNA damage and ultimately cell death in the cancer cells. Moreover, SETD2 loss increased the lethal effect of penometastate, a drug that is currently in clinical development for treatment of leukemia patients with MLL fusions. These experiments might pave the way for a more effective therapy in the future using a combination of compounds. Scientists were able to identify common conserved molecular mechanisms that drive oncogenesis in the context of the large number of different MLL fusion proteins by characterizing the protein-protein interaction networks of distantly related MLL fusion proteins. Their results were now published in Nature Communications. Mutations in the MLL mixed lineage leukemia gene are of interest as they are frequent in AML. They are caused by rearrangements of big chunks of DNA, so-called chromosomal translocations, which can lead to the fusion of 2 otherwise separated genes. In the case of MLL, gene fusions with more than 75% partner genes were found. Above that, it was known that MLL fusion proteins act in the context of large protein complexes which need specific effector proteins to exert their oncogenic function. Ideal targets for a precise therapy, however, it was not clear whether such critical effectors are common for all MLL fusion proteins. AML is not a single disease. It is a group of leukemias that develop in the bone marrow from progenitors of specialized blood cells, the so-called myeloid cells, rapidly growing and dividing. These aberrant cells crowd the bone marrow and bloodstream, which can be fatal within weeks or months if the disease is left untreated. Myeloid cells of various types and stages can become cancerous and cause AML, which makes the condition very heterogeneous and difficult to treat. Thus, finding drug targets that affect as many forms of AML as possible is a prime goal for researchers. Good day, this is Professor Will, CBMD, PhD. Today is Saturday, May 26, 2018. It is 15:54 Eastern Standard Time. A new Achilles heel of blood cancer. New potential target identified to fight acute myeloid leukemia. Date May 18, 2018. Source: Center of Molecular Medicine of Australian Academy of Sciences. Summary: acute myeloid leukemia is an aggressive form of blood cancer. The disease cells often carry mutated forms of specific gene, which is known to function within large protein networks. Applying a combination of proteomic and genomic screens, researchers identified a protein crucial for the survival of the cancer cells. When this potential is lost, AML cells stop growing and accumulate DNA damage, which makes them more sensitive to anti-cancer drugs. In the living xenografts from Beauty, Mayo investigators found that when DNA methyltransferase proteins were present, decitabine showed an effect in triple-negative breast cancer at a low therapeutic dose. The low doses would result in less toxicity and might allow the drug to be used for a longer time, all of which might help to achieve greater therapeutic efficiency. Patients whose tumor does not respond well to chemotherapy are known to be significantly increased risk of recurrent breast cancer and death. Therefore, our focus is to identify new treatment options for these patients. There is a great need to identify additional treatment options for triple-negative breast cancer, which is one of the most difficult to treat subtypes of breast cancer. The study is a demonstration that we can take advantage of many existing FDA-approved drugs to expand their usage by better understanding the mechanisms of how they work and applying them to other cancers. In this study, Mayo investigators identified that the drug 5-AZA2 transpose decitabine, which is FDA approved for the treatment of certain hematological blood cancers, could significantly inhibit the growth of triple-negative breast cancers, and importantly, this effect was also seen in tumors resistant to chemotherapy. This response was dependent on the presence of certain critical proteins called DNA methyltransferase proteins that are present in only a subset of triple-negative breast cancers. This provides a way to identify which patients could benefit from this therapy. Today, this is Professor Will, CBMD, PhD. Today's Saturday, May 26, 2018. It is 15:16 Eastern Standard Time. Science News: new potential treatment option for triple-negative breast cancer. Date May 21, 2018. Source: Mayo Clinic. Summary: in a recent study, researchers identified that the FDA drug approved for myelodysplastic syndrome may be useful to treat triple-negative breast cancer, which is one of the most aggressive and lethal types of breast cancer. This process release describes a basic research finding. Basic research increases our understanding of human behavior and biology, which is foundational in advancing new and better ways to prevent, diagnose, and treat disease. Science is an unpredictable and incremental process. Each research advance builds on past discoveries, often in unexpected ways. Most clinical advances would not be possible without the knowledge of fundamental basic research. Scientists explain that while many studies have shown an association between gut bacteria and immune response, this study is significant in that it identifies not just a correlation but a complete mechanism of how bacteria affect the immune response in the liver. In the same study, the researchers found that bile acids also control the expression of CXCL16 protein in the liver of humans, and wrote that though these results are preliminary, the novel mechanisms described in the study could potentially apply to cancer patients. We asked ourselves, why do mice treated with antibiotics have more CXCL16 production in these endothelial cells? That was the critical point when we found that bile acids control the expression of CXCL16. We then did further studies and found that if we treat mice with bile acids, we can actually change the number of NKT cells in the liver, and thereby the number of tumors in the liver. The investigators next studied the immune cells in the liver to understand how the depletion of the gut bacteria suppressed tumor growth in the liver of the antibiotic-treated mice. Antibiotic treatment increased the numbers of type of immune cells called NKT cells in the livers of the mice. Furthermore, experiments showed that in all 3 mouse models, the reduction in liver tumor growth that resulted from antibiotic treatment was depleted on these NKT cells. Next, they found that the accumulation of NKT cells in the liver resulted from an increase in the expression of the protein called CXCL16 on cells that line inside the capillaries in the liver. To investigate whether gut bacteria affect the development of tumors in the liver, scientists carried out a series of experiments with mice. They used 3 mouse liver cancer models and found that when they depleted gut bacteria using an antibiotic cocktail, the mice that had the antibiotics developed fewer and smaller liver tumors and had reduced metastasis to the liver. The microbiome is the collection of bacteria and other microorganisms that live in or on the body. In humans, the greatest proportion of the body's total microbiome is in the gut, despite extensive research into the relationship between the gut microbiome and cancer. The role of the gut bacteria in the formation of liver cancer has remained poorly understood. What we found using different tumor models is that if you treat mice with antibiotics and therapy depletes certain bacteria, you can change the composition of immune cells of the liver, affecting tumor growth in the liver. This is a great example of how what we learn from basic research can give us insight into cancer and possible treatments. This is Professor Will, CBMD, PhD. Today's Saturday, May 26, 2018. It is 14:10 Eastern Standard Time. Science News: Gut microbiome can control anti-tumor immune function in liver. Date May 24, 2018. Source: NIH, National Cancer Institute. Summary: scientists have found a connection between bacteria in the gut and anti-tumor immune response in the liver. Bacteria found in the gut of mice affect the liver's anti-tumor immune function. The findings have implications for understanding the mechanism that lead to a liver cancer and for therapeutical approaches to treat them. There is an abundance of evidence that the mechanism cancer cells use to defend against oxidative stress could be exploited to enhance the sensitivity of tumors to existing therapies. In the case of TRPA1, inhibiting its activity blocks pain, which is almost always a good thing, and our study highlights its potential clinical utility for cancer treatment. Subsequent studies in cell and animal models have supported this observation. In humans, large-scale clinical trials of antioxidant dietary supplements have found evidence that supplements are beneficial for cancer, and in some cases have suggested that their use may worsen outcomes. Further research is needed to better understand the effects of dietary antioxidants in cancer patients, and the National Cancer Institute recommends that such supplements be used with caution. Buyer beware. The study findings also add to a growing body of evidence that defense against oxidative stress is important to cancer progression. Previous research was among the first to show that antioxidants help cancer cells survive high ROS conditions, particularly when cells can travel away from the original tumor site, as in the case with metastasis. In lung cancer, for example, radiation is an important primary therapy that kills cancer cells by flooding them with ROS. But cancer cells expressing TRPA1 may be able to tolerate radiotherapy better. Inhibiting TRPA1 activity and decreasing this buffering ability might make TRPA1 positive tumors more vulnerable to radiation. We think this could be a possible first use scenario if indeed good TRPA1 inhibitors are developed. Because of its central role in pain sensation and asthma, numerous research efforts and clinical trials are underway to develop small molecules that can safely and effectively inhibit TRPA1 activity. Taken together, the results show that TRPA1 could be an enticing target for therapies against cancer. In mice with transplanted human breast cancer tumors, the researchers showed that blocking TRPA1 with drugs slowed tumor growth. Inhibition of TRPA1 also increased the sensitivity of cancer cells to chemotherapy agents that kill tumors by encouraging programmed cell death. When given together, TRPA1 blockers and chemotherapy significantly reduce tumor size. The cellular program that increases production of antioxidants also simultaneously activates the unique TRPA1-dependent defense mechanism, which allows cancer cells to tolerate enhanced levels of ROS. These two very different pathways work together to help tumors survive and adapt to oxidative stress. When exposed to high levels of ROS, cancer cells appear to rely on NRF2 to activate antioxidant production and to prevent programmed cell death through TRPA1. Antioxidant complement. The team's analysis revealed that an influx of calcium ions through TRPA1 triggers a cascade of cellular signals that suppress programmed cell death. They found that TRPA1 expression is induced by NRF2, a transcription factor that controls the production of antioxidants. They found that ROS levels were substantially higher inside the spiroids compared to the surface, but this disparity had little effect on the survival. When TRPA1 was blocked, however, cancer cells in the interior of the spiroids began to die and were cleared within a few days. Colleagues showed that ROS can activate TRPA1 in neurons. To investigate this connection in cancer, colleagues grew human breast and lung cancer cells under laboratory conditions in 3D spiroids that mimic a tumor's natural structure. Damage prevention. Virtually every cell uses oxygen to power the metabolic process necessary for life, but metabolism also generates ROS, oxygen-based chemicals that are highly reactive and potentially harmful to cellular structures. ROS must be carefully managed to prevent cell damage or death. This is especially important for cancer cells, which accumulate large amounts of ROS as they multiply and metastasized beyond control. Experiments that blocked TRPA1 activity in mice curbed tumor growth and made cancer cells more vulnerable to chemotherapy. Drugs that target TRPA1 are being developed, with some entering clinical trials for a variety of conditions including asthma and pain. Our results suggest that TRPA1 could be a promising target for new treatments against cancer as well. They found that TRPA1 expression is regulated by a cellular pathway that also produces antioxidant compounds that neutralize ROS. Antioxidants and TRPA1 appear to complement each other to increase tumor survival, according to the authors. This was quite an unexpected finding. Tumor cells appear to have co-opt this neuroprotein channel, which is associated with multiple types of cancer, to protect themselves against oxidative stress. The team's analysis revealed that TRPA1 allows cancer cells to tolerate elevated ROS by inhibiting programmed self-destruction, which severely damage our stress cells normally undergo. Certain cancers express unusually high levels of TRPA1, particularly lung and breast cancers, but why they do so has thus far remained unclear. Now, in a new study published online in Cancer Cell on May 24th, Harvard Medical School researchers show that tumor cells use TRPA1 as a unique defense mechanism against reactive oxygen species, ROS, toxic byproducts of cell metabolism. Found primarily in sensory neurons, TRPA1 is a sensor for environmental irritants and has been widely studied for its roles in detecting sensations such as pain and cold in an airway, inflammation conditions such as asthma. Anyone who's taken a bite of a sandwich with too much spicy mustard or a piece of sushi with too much wasabi can attest to the terror-inducing sensation those condiments can cause. These loud warnings to the nervous system of exposure to potentially harmful chemicals are triggered by TRPA1, a calcium channel protein sometimes referred to as the wasabi receptor. Today's is Professor Will, CBMD, PhD. Today's Saturday, May 26, 2018 is 13:44 Eastern Standard Time. Cancer cell co-op pain sensing wasabi receptor to survive oxidative stress. Antioxidants and the TRPA1 channel protein complement each other to increase tumor survival. Date May 24, 2018. Source Harvard Medical School. Summary. Some cancers express unusually high levels of neurocalcium channel known as the wasabi receptor, which plays a role in detecting pain, cold, and other sensations. New research finds cancer cells co-opt this neural channel to increase their tolerance against toxic oxidative stress. Blocking the activity of this channel in mice curbs tumor growth and makes cancer cells more sensitive to chemotherapy. They found that the loss of SFPQ protein was consistent across the board, whether they looked at cells, mouse models, or post-mortem tissue, confirming that they had discovered an important molecular hallmark of ALS. Now that we know these key events are linked to motor neuron death in people with ALS, we can start to think about how we could develop new ways to detect and treat the disease. Cracking the case. The team had uncovered these molecular hallmarks inside the human stem cell models of hereditary ALS. They next confirmed that animal models of hereditary ALS also shared the same features. But to see if the same events could explain non-hereditary forms of the disease, they looked at post-mortem spinal cord tissue from patients. The analysis unearthed what was going wrong in ALS motor neurons. Parts of the RNA sequence that don't code for proteins are usually cut out before the RNA is translated into protein, but in the ALS motor neurons this wasn't happening as effectively. This guided the team to collectively discover that a protein called SFPQ, which normally resides inside the cell nucleus, was in fact leaving the nucleus in diseased motor neurons. Initially using conventional analysis, we didn't detect any differences in RNA sequencing between healthy and diseased motor neurons. But we knew something must have been going wrong to make the ALS motor neurons die. So we wrote a whole new program to dig deeper into a genetic code, and when the results came back, we knew we were onto something. Digging for treasure, armed with a whole load of RNA sequencing data from healthy and diseased motor neurons at different stages of disease. To drill down into the data and work out exactly what was going wrong. Bioinformaticians are highly skilled scientists who developed advanced computational techniques to study biological data. Turning back the clock, using cutting-edge stem cell technology, scientists in Ricky's lab took skin cells from healthy volunteers and patients with ALS and turned them into stem cells capable of becoming many other cell types. Then, using specific chemical signals, they guided the stem cells into becoming motor neurons that they could study in the lab. By turning back the clock, we could watch what happened to the motor neurons over time to lead to the disease. But even with RNA experts on board, comparing RNA sequencing in healthy and diseased motor neurons couldn't provide the full picture. The suspect. Previous studies had implicated deregulation of RNA, a molecule closely related to DNA that has a vital role in coding, decoding, regulating, and expressing genes. In ALS, for instance, patients with a hereditary form of ALS often have genetic mutations that prevent their RNA from functioning properly. It is a really devastating disease. Patients progressively lose the ability to move, eat, speak, and ultimately breathe. We set out to uncover the molecular events that lead to ALS in the hope that one day we can develop new treatments for patients. The mystery. 3 years ago, a group of clinical neurologists, molecular biologists, computer scientists from different London institutes decided to work together to solve the mystery of why motor neurons die in patients with amyotrophic lateral sclerosis, ALS, also known as motor neuron disease. But it isn't always easy for researchers to work together. Finding the right people to collaborate with can be tricky, especially when some are understandably protective of their ideas. Then there is the practical challenge of meeting up, exchanging ideas, and carrying out the research miles and even time zones apart. This is Professor Will, CBMD, PhD, today, Saturday, May 26, 2018. It is 20:31 Eastern Standard Time. Why motor neurons die in patients with motor neuron disease. Date, May 25, 2018. Source, the Francis Crick Institute. Summary, a group of clinical neurologists, molecular biologists, and computer scientists have worked together to solve the mystery of why motor neurons die in patients with motor neuron disease. 19th century research identified microbes as agents of disease and set the stage for 20th century breakthroughs in antibiotic therapies. But antibiotic resistance is now a global crisis, and we are also now aware that antimicrobials can harm beneficial species too. And as with natural habitats where successful restoration techniques will vary from year to year and place to place depending on individual circumstances, gut restoration requires a similar approach, one toward personalized medicine, given that no two gut sets of gut microbiota are identical, except for that they are all essential to the person's health and all subject to disturbances, including the food people eat and the medicine they take. All of that inspired me to think about ways that my knowledge of restoration ecology might help to guide physicians away from treating and misdiagnosing other people the way that they had treated and misdiagnosed me. Gut microbes perform a wide range of beneficial functions. Among other things, they produce nutrients in the form of short-chain fatty acids and vitamins, control blood sugar and weight, reduce inflammation, and even improve mental health and psychological well-being. I was diagnosed with Crohn's disease and put on a monoclonal antibody inhibitor for life, except that I cleaned up my diet, stopped getting the gut irritation, cut out the monoclonal antibody inhibitor, and my symptoms are gone. 2 doctors overturned the Crohn's disease diagnosis in writing. One tenet of restoration ecology that is ignored by Western medicine is that active interventions will not succeed if a passive platform is not established. The disturbances has to be removed. Part of what compelled to look at the human microbiota from an ecological perspective was the chronic gut irritation he developed from years of doing research in the tropics and taking antibiotics for bugs picked up at unsanitary field stations. Restoration techniques fall into one of 2 categories, passive and active. Passive means removing whatever disturbances have harmed an ecosystem, such as an unhealthy diet, and then letting the ecosystem heal itself. If that doesn't work, the next step is active restoration in nature that could mean physical manipulation of a landscape, exterminating unwanted species and introducing desired ones. In the gut, it might involve probiotics, antibiotics, and fecal microbiota transplants. The highest known cell density of the microbial habitat on the planet is found in the human gut. There can be more than 1,000 species of bacteria containing more than 500,000 genes. But applying techniques of ecological restoration, assisting the recovery in an ecosystem that's been degraded, damaged, or destroyed, to the field of medicine has been a largely unexplored option. A paper published in the Coordinary Review of Biology notes that throughout history humans have harmed beneficial species, often inadvertently through advances of culture and technology. Affected species include those inside of people as well as around them. Western doctors generally ignore diet and chronic disease, even diseases of the gut. To do this research in part by his own health colleagues, they do not overly encourage or support their patients to change their diet away from high-fat and high-sugar industry and policy have created a platform for people to eat terribly in this country, and many Americans do eat terribly. That means drawing on techniques which had been developed by scientists over the last half-century that have restored species diversity and ecosystem function of natural habitats. Scientists describe the restoration ecology approach toward patient health. Every person is like an ecosystem, he says. And effectively fighting chronic disease requires fostering the communities of symbiotic gut microbes that people need for their health.